Hello everyone, and welcome to today's The Scientist webinar. I'm Betsy Young, Technical Editor for The Scientist, and I'll be moderating our discussion. Today, our panel of experts will be discussing the editability of the epigenome. Between the endogenous cellular repair machinery and erasing enzymes, the epigenome is an ever-changing code. Will learning to rewrite the epigenome hold the key to better understanding life itself? Our panelists will weigh in. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the panelists will address these during the Q&A session following the presentations. To ask a question, simply type your query into the question box located on the bottom left-hand side of your screen. We will try to address as many of these questions as we can during our Q&A session. The webinar platform is user-friendly. You can move or resize any of the windows simply by stretching them at the bottom right-hand corner. You may need to move or minimize some of the windows to see the live view. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. BioLegend develops and manufactures highly recognized world-class antibodies and reagents at an outstanding value to customers for biomedical research. Our broad product portfolio includes flow cytometry, cell biology, and biofunctional molecules for research in immunology, neuroscience, cancer, cell biology, stem cells, and more. Our aggressive product development program, accomplished through technology licensing collaborations and internal research and development, has produced a product offering of over 17,000 products, which have been collectively cited in over 25,000 peer-reviewed journals. BioLegend also offers a wide range of custom services, including assay development, sample testing, and conjugation. BioLegend's reagents are supported by superior customer service and a quality management system dedicated to continuous improvement that is certified for ISO 9001-2008 and ISO 13485-2003. Innovation is at the heart of biotics engineering. Our liquid handling product development starts with questions like, how can we create a product that enables researchers to achieve more accurate and precise results? How can we use less resources? How can we drive this innovation across our industry and beyond to make a significant and overwhelmingly positive impact? We invite you to join our quest for game-changing innovation, one aspiration at a time. Because your data quality matters, your pipette tips matter. Find out more at www.biotics.com. Active Motif is the industry leader in developing and delivering innovative tools to enable epigenetics and gene regulation research for the life science community. We have an extensive range of assays, validated antibodies, and genome-wide services for use in a variety of applications, including ChIP-seq, histone characterization, and DNA methylation analysis. Zymo Research Corporation, also known as the Epigenetics Company, is a privately held company based in Irvine, California. Since its inception in 1994, it has been serving the academic and biopharmaceutical scientific communities by providing DNA and RNA purification products. Zymo Research was an early adopter in the field of epigenetics and offers a line of products used in DNA methylation detection and quantitation. In addition to the epigenetic and nucleic acid purification products, Zymo also provides genetic, epigenetic, and transcriptome analysis services. For more information, please visit www.zymoresearch.com/epigenetics. Follow Zymo Research Corp on Facebook, LinkedIn, Twitter, and Instagram. And with that, let me introduce our first speaker, Alex Drohat. Dr. Drohat received a PhD in biochemistry and molecular biology focused on structural biology and NMR from the University of Maryland School of Medicine. He then went on to conduct a postdoctoral research at Johns Hopkins University and the University of Maryland Center for Advanced Research in Biotechnology, where he continued to develop an interest in structural biology, but focused on enzymology and DNA repair. That brings us to the work he's conducting as an associate professor in the Department of Biochemistry and Molecular Biology at the University of Maryland School of Medicine. His research deals primarily with the enzymes responsible for identifying and repairing mismatched DNA as a part of the base excision repair pathway, DNA glycosylases. He and his team have studied the mechanisms behind active demethylation of DNA, and he will discuss these findings today. Dr. Drohat?
Well, thank you, Betsy, for that introduction and for your kind invitation to share our work with you today. So we'll be talking about erasure of epigenetic marks in DNA. And of course, there are epigenetic marks in general can include modification to histones, and we'll focus only on DNA modifications and their erasure, and specifically focus on the erasure of 5-methylcytosine or derivatives of that base, as we'll see more about in just a few minutes. And so just by way of introduction, 5-methylcytosine is, of course, the most abundant DNA modification found in all three forms of life. It's really important, of course, for regulating gene expression, critical for proper cell differentiation, development, genome integrity, among other important processes. Methylation patterns, it turns out, can vary or do vary during normal embryonic development. And it's been observed in many different types of cancer that aberrant methylation patterns are contributing to cancer and also to other diseases, including neurodegenerative diseases. And so the question of how 5-methylcytosine can be erased from DNA is one that's been open and under investigation for quite a few years. It's well known, of course, how the methylcytosine mark is generated in the first place through DNA methyltransferases. But the question of reversing this mark has been harder to establish in terms of how this occurs. And so we'll talk about a couple of different possibilities and we'll think about two broad approaches for this process. And so the first, of course, is passive DNA demethylation. And so, and for the purposes of my talk today, I'll talk about, I'll refer to this as processes that involve DNA replication. So, of course, 5-methylcytosine can be diluted through passive dilution simply through replication without maintenance methylation. And it's also true that oxidized derivatives of 5-methyl-C can also be diluted in the same way through DNA replication. But today I want to focus on mechanisms for active DNA demethylation. And by that I'm referring to processes that are completely independent of DNA replication. Okay, and so one might imagine that the most straightforward way for demethylating DNA would be simply to cleave the methyl group directly off of the 5-methylcytosine base. And so this is an idea that actually was proposed a few years ago, back in around 2000, I believe. But this initial report suggested that a protein known as MBD2 could directly cleave the methyl group from 5-methylcytosine. But it turns out that this initial report, these initial findings, have not been reproduced by other groups. And at this point, there's no known enzyme that can directly excise the methyl group from 5-methyl-C. And so it turns out that studies over the past decade or so have shown that erasure of 5-methyl-C requires excision of the entire 5-methylcytosine base or a derivative of 5-methylcytosine involving DNA repair enzymes. And specifically, these enzymes are those involved in DNA base excision repair. And so I'll just give a brief overview of this pathway. And this will be one version of this pathway. There are a number of different versions that are found in cells, but this will give us just an overview of the basic steps of base excision repair that are important when we think about active DNA demethylation. And so it turns out the first step in this pathway is catalyzed by a family of enzymes known as DNA glycosylases. And so these enzymes use, in general, a base flipping mechanism to flip a target base out of the DNA duplex into an active site. Recognize that this is a substrate base, cleave the bond connecting the base and the sugar, and that generates an A-basic site in the DNA. The next step in the pathway typically involves an AP endonuclease. And so these enzymes recognize the A-basic site that is generated by the DNA glycosylase, and they nick the DNA on the 5' side of that A-basic site, generating the 3' hydroxyl group that's needed for the following step that's catalyzed by a DNA polymerase. Okay, and then excision of the remnant of the A-basic nucleotide can be conducted by the DNA polymerase or perhaps other enzymes. And then finally, the nick in the DNA is sealed by a DNA ligase. So the key first step here in this pathway, and the step that we'll focus most on as we talk about mechanisms of DNA demethylation, is that conducted by the DNA glycosylase. So excision of the modified base, or in some cases a mismatched base in DNA. And so if we think about how base excision repair might mediate active DNA demethylation, the obvious first choice might be to have a DNA glycosylase that can simply recognize 5-methylcytosine and cleave that base entirely from DNA, generating an A-basic site, and then follow-on enzymes come in to restore cytosine at that position. And so this model has actually been proposed for both animals and plants, and we'll cover in just one slide here what happens in plants, and then move on to animals afterwards. And so it turns out that in plants there are DNA glycosylases that do recognize 5-methylcytosine, cleave the bond, and follow-on steps in base excision repair then restore cytosine. It turns out these enzymes in plants, these glycosylases are bifunctional, so they also have lyase activity. They can nick the DNA backbone. So a little bit different from the pathway that I showed a couple of slides back. But nevertheless, these are enzymes that recognize 5-methyl-C, remove it from the DNA, and follow on base excision repair enzymes and come in to restore cytosine. And the 4 known glycosylases in plants are shown at the bottom of the slide. And I think Dr. Hong may speak a little bit more detail to the process of DNA demethylation in plants. So I'll just leave it at that for how things happen in plants, and move on to how this process might occur in animal cells. And so, as I mentioned, previous studies had suggested the possibility that a similar process occurs in animals. That is, that a DNA glycosylase directly removes 5-methylcytosine from DNA. And this had been proposed for 2 enzymes that we'll talk about in a little bit more detail in a few minutes, thymine DNA glycosylase and methyl binding domain 4. But it turns out that subsequent studies have shown that neither of these enzymes has efficient activity for excision of 5-methylcytosine directly. And it also it turns out that none of the glycosylases that are found in plants have been identified in animal cells. And so the short, the summary there is that glycosylases in animals are unable to remove 5-methylcytosine directly. And so studies over the past decade or so have shown that what happens in animal cells is that 5-methylcytosine is modified to give a version of the base that can be recognized and excised by a DNA glycosylase, and thereby DNA base excision repair can ultimately restore cytosine in the spot where 5-methylcytosine had been found. Okay, and so one of the first proposed mechanisms for how this might occur involved a process known as active deamination. And so the idea here is that the 5-methylcytosine base is deaminated to give thymine, right? So this converts a methyl C guanine, a normal base pair, to a TG or a GT mispair. And so it was proposed that either the AID or the ABO-Beck enzymes can mediate this active deamination to give a GT mispair. And so there are two enzymes found in mammalian cells that are well known to have activity for GT mismatches. And these are TDG and MBD4. And so these enzymes selectively remove the thymine from a GT mismatch, generating an A basic site, and subsequent base excision repair then restores a GC base pair. And so this was one of the early proposals for how DNA demethylation could occur in mammalian cells. But it turns out that recent studies have shown that the AID APO-Beck enzymes in fact have relatively weak activity for acting on methyl C. These enzymes act preferentially on cytosine and on cytosine in single-stranded DNA and have specificity for certain DNA sequence contexts. And so together these observations indicate that the deamination pathway is probably not an efficient pathway for active DNA demethylation. Now, another potential concern or argument against this pathway is that this pathway is going to, of course, generate GT mispairs that are mutagenic. So if these mispairs don't get processed by TDG or MBD4 and subsequent base excision repair, if they don't get processed prior to DNA replication, then these mismatches can lead to a mutation in DNA. So this is considered a fairly risky approach for active DNA demethylation. It also turns out that these glycosylases, the GT glycosylases, the activity for GT mispairs is relatively inefficient, and this might be governed, this might be due to the fact that these enzymes have to play a kind of a balancing act between efficient activity for GT mispairs, where they're removing the thymine, but at the same time avoiding any activity on the vast excess of AT base pairs. And so their activity might be governed by this balancing act. And a number of studies have suggested that this relatively inefficient activity of these enzymes contributes to the pretty high number of C to T transitions that are observed at CPG sites in genetic disease and cancer. And so that's another argument, in my opinion at least, against the process of using deamination to create what's really a mutagenic lesion for the process of active DNA demethylation. But additional studies are needed to really to learn more about the potential role of this pathway. in active DNA demethylation. Okay, so another pathway that has recently come to light was initiated or first imagined by the seminal discovery that TET enzymes can oxidize 5-methyl-C to give hydroxymethyl-C. And so this was discovered in 2009. Related enzymes that can convert thymine to 5-hydroxymethyl-uracil and then on to formal uracil and carboxyl uracil suggested that these TET enzymes could also catalyze 2 additional modifications. And so it turns out that these additional activities were discovered in 2011. And so the TET enzymes can generate 3 different derivatives of methyl-C. So the first being hydroxymethyl-C. They can then oxidize that to give formal cytosine, and then further oxidize formal C to give carboxyl C. And so these are now well-established epigenetic modifications that are found in DNA. And with regard to the question of DNA demethylation, an important finding was that thymine DNA glycosylase, the enzyme we mentioned a couple of slides ago for GT mismatch activity, this enzyme can very efficiently remove both formal cytosine and carboxyl cytosine from DNA. Again, giving an A basic site, and then subsequent base excision repair enzymes come in to generate cytosine. So a multi-step, multi-enzyme pathway for active DNA demethylation, starting with methyl-C, oxidation by the TET enzymes to give either formal cytosine or carboxyl cytosine, excision of one of those bases by TDG, and then follow-on base excision repair. And so this is really the only pathway that's been established both in vitro and in mammalian cells for efficient active DNA demethylation. And shown at the bottom is just some of our data collected for TDG, showing very efficient excision of both formal cytosine and carboxyl cytosine. And when I say efficient, it's more efficient than the GT activity for FC excision, and roughly the same for CAC and activity versus GT mismatch activity. So an enzyme TDG that had been long recognized for excision of thymine from GT mispairs, now shown to have very efficient activity for excision of FC and CAC. Now, another way that TDG had been suggested to play a role in active DNA demethylation involves both oxidation and then subsequent deamination of the methyl-C base. Right? So the first step being oxidation to give hydroxymethyl-C, and then the idea that this 5-hydroxymethyl-C could be deaminated to give hydroxymethyl-uracil. Right? So this would generate an HMU-G mismatch in the DNA. And there are 3 enzymes that are known to have efficient activity for excising HMU from these mismatches, and those include TDG, the enzyme we just talked about, as well as SMUG and MBD4. But again, studies that have been done in vitro on AID-ApoBec enzymes indicate very weak activity for HMC, so even weaker than observed for methylcytosine. And again, these enzymes are really very efficient for deamination of cytosine to give uracil, but not efficient for deamination of HMC to HMU. And another point to note is that this is, again, a potentially risky endeavor, right, because the creation of an HMU-G mispair could lead to mutations if that mismatch is not handled by one of the base excision repair glycosylases prior to DNA replication. Okay, so jumping back to TDG and the TET initiated pathway, it's also important to note that studies reported in 2011 find that TDG is essential for embryonic development in mice. And so this is the first DNA glycosylase that has been found to have this phenotype upon depletion. Although these initial studies suggested that this might relate to activity of TDG either on a GT mismatch created through active deamination, or perhaps on an HMU guanine mismatch generated by TET activity and subsequent deamination. The most likely explanation at this point for the embryonic lethality of TDG is its role in demethylation by removal of FC and CAC. So initial steps initiated by the TET enzymes creating methyl C and then oxidation onto FC and CAC. And I should mention that both of the studies showed that not only the presence of the TDG protein, but its catalytic activity in particular is essential for embryonic development. So the glycosylase activity, base excision activity, is essential for development. And again, it seems most likely at this point that phenotype reflects the need for excision of FC or CAC during active DNA demethylation. And so with these findings, we've been interested in understanding how it is that TDG can excise these two bases, formal cytosine and carboxyl cytosine, while at the same time avoiding activity on cytosine, methyl C, and HMC. And again, this TDG is an enzyme that's long been known for acting on GT mismatches, and that's an activity that we've been studying for over a decade at this point. And we've also, with the new findings that it works also on FC and CAC, have also been focusing on its specificity for those two bases. So we take a combined biochemical and structural biological approach to these studies. And just by way of a brief introduction, I just want to kind of review how it is that TDG excises bases in general. And so shown here is a stepwise pathway for base excision. And so this is a pathway or a mechanism that's been shown for other DNA glycosylases, hasn't been established for TDG, but based on the findings of these other enzymes and on non-enzymatic reactions, it seems likely that TDG follows this stepwise mechanism. And the important point here is that in this process, the first step is cleavage of the N-glycosyl bond, the base sugar bond connecting the base and the sugar. And this generates a very unstable, short-lived intermediate. And so as you can see, when that bond is cleaved, negative charge migrates up into the departing base. And so a key feature of TDG and other DNA glycosylases is to stabilize that negative charge in the first transition state and in this intermediate. And so it turns out that studies we did on TDG back in 2006 showed that there's a very clear and linear dependence of its activity for a variety of modified cytosines and uracils, a dependence of its activity on the leaving group quality of that departing base or the cleaved base. And so we'll see in a few minutes why this is relevant for excision of both FC and CAC. But the take-home message here is that that the activity of TDG, the specificity of TDG, depends, at least in part, on the leaving group quality of the base, of the target base, once that glycosylic bond is cleaved. We also take a structural approach, and we have solved a number of structures of DNA-bound TDG. And this slide just illustrates the idea that we can solve structures for either the enzyme substrate complex, where TDG binds and it flips a target base into its active site. And so we can capture that snapshot with the base flipped into the active site, but the base not being cleaved by the enzyme. So this gives us an idea of how the enzyme recognizes various bases. We've also solved crystal structures of enzyme product complexes of TDG bound to abasic DNA. Now, of course, the intermediates in this pathway are just too reactive and too short-lived to be captured by crystal structures. So we'll start with excision of FC and how this occurs by TDG. And so here's a crystal structure of the enzyme. So the enzyme residues are shown in white, and the strand that contains FC is shown in yellow. And you can see that we've got a number of important hydrogen bond interactions. I should just mention that this structure was made possible by the use of a 2' fluoro analog. So placing a 2' a fluorine at the 2' position allows for base flipping, so we can see the enzyme substrate interactions, but it prevents cleavage of the bond, the base sugar bond by the enzyme, so giving us a nice approach for obtaining an enzyme substrate structure. So a number of mutagenesis studies have shown only a minor role for active site side chains that are near the formal cytosine base that's flipped into the active site. And it appears that really backbone interactions with a number of different amide groups are important for stabilizing negative charge that migrates out onto the FC base when that bond is cleaved. So upon, if we look at the mechanism on the lower right, upon cleavage of that bond, that negative charge moves into the base, and it can resonate through delocalization out to the O2 and the formal oxygen. And we see in the crystal structure that the enzyme backbone and a water molecule form interactions with both of these groups, both the O2 and the formal oxygen. Now, it turns out that FC is an inherently good leaving group. The formal group turns out to be electron withdrawing, so you might kind of think of it as pre-activated for excision. But of course, these interactions provided by the enzyme are also important for stabilizing those negative charges for the cleaved base, but also most likely for recognizing formal C once it flips into the active site prior to base excision. Another key feature of activity for this enzyme is for binding the nucleophilic water molecule. And we see in this crystal structure on the left the mechanism by which TDG binds the nucleophilic water molecule. I mentioned a few slides back that the catalytic activity of TDG is essential for embryonic development. And so the residue that was knocked out to selectively remove the catalytic activity while enabling the substrate to still bind to substrate is this N140 side chain. And so that's a side chain that's absolutely essential for catalysis. Most likely, its primary role is to bind and position that water molecule. Threonine 197 also plays an important role in that regard. Okay, so that's a snapshot of how things work for formal cytosine excision. And what about carboxyl cytosine? So here things get a little more complicated. And the reason for this is that under physiological conditions, carboxyl cytosine exists as an anionic base. And that's due to the negative charge on the carboxyl substituent. Now, although you'll see in a number of papers the carboxyl group drawn with a proton attached, right? So a proton or a protonated carboxyl group. A number of studies from our lab and others have demonstrated that under physiological conditions, this carboxyl group is negatively charged. And so the implication of that for TDG excision is that most likely it cannot excise CAC as it's found in DNA. And the reason is that it's just a very poor leaving group, right? So if you've got a base that already contains a negative charge and you try to push a second negative charge into that leaving group, then it's just most likely not going to happen. And so it turns out that for CAC, unlike formal cytosine, TDG needs to protonate the CAC base prior to bond cleavage. And as shown here on the bottom left is one potential form of protonated CAC. It's also possible that it gets protonated on the carboxyl group itself, and additional studies are needed to establish that in detail. But nevertheless, studies of the pH dependence of TDG activity show conclusively that that the excision of CAC follows an acid-catalyzed mechanism. So you can see that there's a very strong dependence of CAC activity on pH, where activity increases with decreasing pH. By contrast, we see no pH dependence for excision of Fc from DNA. And we can fit this data to find that the CAC base is protonated when it's flipped into the TDG active site with a pKa of 5.8. And so here's a crystal structure that was solved by another group a few years ago with CAC flipped into the active site. And what we see is that it forms a number of similar interactions with the backbone compared to those that we saw for formal cytosine, including, for example, the interactions with the O2 of CAC and the backbone groups 139 and 140. We're in the process of solving some higher resolution structures with CAC flipped into the active site, and these structures will show water molecules, and which is important because the protonation of CAC in the active site most likely involves a proton from solvent rather than a general acid of the enzyme, because there is no residue that would serve such a role. So it turns out we looked at some mutations of this mutational effects for CAC excision. We see that alanine 145, which is kind of pointing towards the carboxyl group, if we convert that to a glycine, we see a pretty big increase in activity, a fourfold increase. simply suggesting that the methyl group of that alanine is creating some level of steric hindrance for flipping of CAC into the active site. We saw similar effects for the activity of TDG on GT mispairs, where the methyl of thymine was clashing to some extent with the methyl of alanine 145. With regard to other side chains, we don't really see a significant contribution for any of the other active side chains that are near the flipped CAC base, except with with regard to asparagine 191. And so strikingly, what we see for 191 is that if we convert that side chain to alanine, we see absolutely no detectable activity for CAC. By contrast, it has normal activity for FC. And if you look at the crystal structure, 191 is kind of up at the top and in the center, and you see that there's a carboxyl oxygen that is contacting the N3 position. Now, the only way that such an interaction could be favorable would be if there is a proton at that N3 position. And so there are two potential protonated forms of CAC that could fill this role. So if CAC is protonated at N3 to give the Zwitter ion or the CAC imino structure, and these are the two structures shown at the bottom right on the outside, those two protonated forms would be consistent with an essential role for N191. But at this point, we've got more studies are needed to really kind of nail down exactly where CAC gets protonated and exactly what the role of this residue is. But I would point out that this N191A variant of TDG is kind of a useful tool for studies of how DNA demethylation occurs in cells, because you've got a variant that has absolutely no activity for CAC, but full activity for excision of FC. Okay, and so with that, I just want to summarize. So studies to date suggest that the TET-TDG base excision repair pathway is the sole pathway for active DNA demethylation that's been established both in vitro and in mammalian cells. We've shown that FC is more or less pre-activated for excision by TDG, which provides key interactions, hydrogen bonds, to two of the oxygens of formal cytosine once it's flipped into the active site. And these are most likely important for both recognition and for cleavage of formal C from DNA. By contrast, CAC needs to be activated in order to be protonated. This occurs through protonation of CAC once it's flipped into the active site. And again, the mutational studies surprisingly revealed an absolutely essential role for 191 in excision of CAC, but not FC, suggesting that this residue may be important for stabilizing a protonated form of CAC. And again, acid catalysis seems to be absolutely required for excision of CAC. Okay, and with that, I want to acknowledge all the people who did the work. And so Swarna, a postdoc in our group, did much of the work on the FC crystal structures. Atanu, a former postdoc in the lab, did much of the biochemical studies and some of the structural work as well. Longtime collaborator Ed Pozarski at the University of Maryland has been helping us with crystallography. Want to also acknowledge Mark Greenberg at Johns Hopkins University, who synthesized the 2' fluoro FC analog that was critical for solving that crystal structure with FC. Dr. Lee at Rutgers University who's been instrumental in helping us with theoretical calculations. That's it. And I want to thank you for your attention, and I'll be happy to take questions in a few minutes.
Thank you, Dr. Drohat. I'd like to take a moment to remind our audience that they can ask questions at any time by simply typing their question into the question box. The panelists will respond to these questions during the Q&A session. Our second speaker today will be Samuel Hong. Dr. Hong received his PhD in biomedical sciences from Emory University, where he joined the laboratory of the renowned structural biologist Xiaodong Cheng to study the basic science of epigenetics. Since graduation, Dr. Hong has been working as a postdoctoral fellow studying the structural mechanism of programmed DNA frameshift events under the mentorship of Christine Dunham. He is currently preparing to begin a new postdoctoral position at MD Anderson Cancer Center in the Department of Lymphoma and Myeloma to expand on his translational research on multiple myeloma. Dr. Hong?
Thank you, Betsy. Can you hear me clearly? I just want to make sure that everyone can hear me.
Yep, you sound great.
Okay, thanks very much. Okay, thank you, Betsy, for organizing the webinar, and I really appreciate the invitation to the panel and for this webinar, as well as the very nice introduction, so thank you very much. It is my privilege to be able to speak to the audience in the Scientist webinar platform. And also special thanks to Etha, who has accommodated my multiple requests to troubleshooting the slides I will be presenting today. And also thanks very much for Dr. Drohat for the very insightful presentation on the mechanism of TDG. And so let me begin my presentation. The past 25 years of studies on proteins that interact with DNA in epigenetic context have yielded a common framework by which a DNA base is modified, recognized, and removed in a single unit basis. So this framework is known as DNA base flipping. And my goal today is to briefly overview what base flipping is, highlight what players are involved, and provide insight as to why it is so pervasive when it comes to epigenetics. Now, when we talk about epigenetics and DNA modifications, we must start with DNA methylation. It is the earliest and the most widely understood modification of DNA. There are at least 3 types of DNA methylation generated by different types of DNA methyltransferases, but cytosine methylation at the carbon 5 position of the ring is the most widely studied one. What I mean by carbon 5 is, in counting the carbons within the pyrimidine as shown on the left, it is the 5th carbon of the ring in that counting as shown that becomes methylated. So if I may briefly go over the reaction mechanism, a cysteine residue within the enzyme catalytic center activates the carbon 6 of cytosine to enable a nucleophilic attack from carbon 5 position towards the methyl group of S-adenosylmethionine, abbreviated as SAM cofactor, in which the methyl group is shown in red color. So that SAM becomes SAW as adenosyl homocysteine after donating this methyl group. Now, if you take a closer look at the second and especially the third step of the reaction, you'll notice that the cysteine residue and the cytosine base form a transient covalent bond. And after forming this covalent complex and transferring of the methyl group, the following elimination reaction is initiated by a basic residue within a catalytic center that attacks this hydrogen on the carbon 5 position, resulting in the release of the enzyme from the base. And this reaction then completes the generation of 5-methylcytosine, or simply 5-MC. And this is the base that we think of the most when we think about DNA methylation. In efforts to understand how this classic epigenetic event of writing on DNA happens, a group of scientists published a structure in Cell in 1994. So what they try to do was they try to capture this intermediate complex where you expect the covalent bond between the enzyme and the target base. And in order to stop the reaction after this covalent bond formation, they used 5-fluorocytosine as substrate, which has fluorine instead of hydrogen in the carbon 5 position of the cytosine. So if you look at the top red box, if you have the fluorine in the carbon 5 position instead of hydrogen there, it'll stop the reaction in this step because the enzyme cannot proceed to the elimination reaction step. And this way, the scientists became successful at being able to capture and solve the structure of bacterial methyltransferase, HAHA1, in complex with DNA containing this substrate. And in that structure, they observed for the first time that the target base is flipped out of the DNA helix by the enzyme during this catalytic reaction. As shown in the bottom right in the red box, a specific cysteine residue, cysteine 81, this enzyme makes a covalent bond with the flipped cytosine, which just received a methyl group from SAM at the carbon 5 position, just as predicted in the proposed reaction mechanism in the 1980s. And in that same position of the carbon 5, you can see the fluorine atom in cyan color. The SAW in blue is in close proximity, and the red arrow indicates the trajectory of the methyl group transfer. So this seminal observation of structural framework of accessing a DNA base by flipping the base was particularly important because it provided, for the first time, the structural basis of how a DNA base that is buried within a double helix can be reached effectively and modified specifically. As I mentioned, there are two other types of DNA methylation, especially in the prokaryotic system. There are adenine N6 methylation and cytosine N4 methylation. The methylation here involves the exocyclic amine group. So it is the methylation of nitrogen instead of carbon, as we normally see. So there's a recent report that Drosophila may utilize adenine methylation in their genome as an epigenetic modification. And we know that in mammalian system, mRNA can become methylated at the adenine N6 position. So we may want to consider these types of methylation as epigenetically significant. But more importantly, there is a point to be made here as to what we can learn from the consequence of methylating the nitrogen atom as opposed to methylating the carbon atom, because it matters when it comes to the event of DNA demethylation. And I'll come back to that point in a moment. But before discussing further on that topic, I would like to mention the structural features of methyltransferases that generate these exocyclic amine group methylation. In the bottom left is the structure of cytosine C5 methyltransferase that I just showed in the previous slide. In the middle is the structure of bacterial adenine N6 methyltransferase, TOC1, in complex with DNA reported in 2001, showing the flipped adenine in green color and SAM analog in blue. What you'll notice is that the protein folds colored in cyan helices and beta sheets in purple look very similar. So there's a conserved MTase fold, methyltransferase fold, preserved among different types of methyltransferase that engage in the transfer of methyl group from SAM to the target substrate. And you can see the same fold for the methyltransferase, for instance, PBU2, that methylates the exocyclic amine group cytosine, as shown on the right. So this MTase fold is conserved in mammalian DNA methyltransferases and across other types of methyltransferases in different species. In fact, histomethyltransferases that methylate the lysine side chain terminal amine group, which are also important in epigenetic modifications, share the similar MTase structural fold because it also utilizes SAM to transfer the methyl group to the target substrate. As for the fate of 5-MC after DNA methylation event, there have been, as Dr. Droyd have mentioned in the presentation, there's been active interest in understanding how DNA demethylation happens. And as you would now be well aware, people have tried to identify DNA demethylase enzyme. But then in 2009, an interesting discovery was made regarding the discovery of a TET 5-MC dioxygenase mentioned in the previous presentation. It is now widely known in the epigenetics community that this TET enzyme can write on 5-MC in the methyl group to generate hydroxymethylcytosine, orbital cytosine, and carboxyl cytosine. As a dioxygenase writer of DNA modification, this enzyme belongs to a completely different class of enzyme as compared to DNA methyltransferases. It utilizes alpha-ketoglutarate as a cofactor, along with iron and oxygen to specifically oxidize a carbon group, highlighted in yellow in this page. But the foundational requirement of the protein for it to be able to access a specific DNA base for a particular chemistry, that principle stands here in the same manner. So the enzyme would harbor a mechanism to selectively recognize the substrate base out of the other options within a variety of sequence contexts across genome for a specific reaction. And lo and behold, after 4 years after discovery, the structure of human TAT enzyme was reported in 2013 in complex with DNA containing 5-MC as shown below. Another eukaryotic TAT enzyme in complex with DNA containing 5-MC or 5-HMC have also been reported around the same time. And all of them consistently show that this enzyme flips the target base for dioxygenase reaction. In the human TAT structure to the bottom left, the green denotes the flipped 5-MC and the blue denotes the alpha-ketoglutarate, with iron shown as an orange ball. The structural fold is characterized by this helices, alpha helices in red color, attached to this yellow data sheet sandwich where the reaction happens. Now the implications of TAT structure extends beyond the fact that it flips the target base. As we can see, because in the bottom, TAT is a structural homolog of AlkB enzyme, which is a DNA repair enzyme that also belongs to the dioxygenase class, which is a base repair enzyme. So as a base repair enzyme, AlkB is known to recognize a base with alkyl damage, such as N-methylation, and it flips the base for a corrective dealkylation reaction. In other words, AlkB is a demethylase. In addition to an apparently methylated substrate, it can also recognize N6-methyladenine to a certain extent, although it is not the most desired substrate in E. coli. But the point is, if you use N6-methyladenine as an example here, AlkB would demethylate the methyl group attached to the primary amine group and the exocyclic amine group of this adenine, methyladenine. And what actually happens during the reaction is a bit more interesting, because AlkB-mediated demethylation involves a transient formation of the hydroxymethyl group, just as the one mediated by TAT enzymes, as shown in the third step of the reaction here. But it is transient because this hydroxymethyl group proceeds to a spontaneous release in the form of formaldehyde. So AlkB does not actively take out the methyl group for demethylation, but instead this enzyme-driven oxidizing event results in a spontaneous release of the oxidized group, and the end result is the demethylation. So this is the most significant characteristic of oxidizing the methyl group attached to the nitrogen versus oxidizing the methyl group attached to the carbon. The latter case does not result in a spontaneous release of the hydroxymethyl group because it is not as labile as the one that's attached to the nitrogen. And as a note, this happens with histone lysine demethylases that also belong to the dioxygenase class utilizing alpha-ketoglutarate. In case of histone, there's a lysine methylation, and it is the terminal amine group of the lysine side chain that gets methylated. When a histone demethylase reacts with this methyl group, it forms a hydroxymethyl group. And that hydroxymethyl group is spontaneously released as a formaldehyde, and that results in histone demethylation. In the case of oxidizing the methyl group in 5-MC, as mentioned, the hydroxymethyl group attached to this carbon is quite stable, allowing for a subsequent oxidative reaction by TAT enzyme to produce 5-FC as well as 5-CAC. So what will be the function of this differently modified basis? The existence of TAT and the product of its activity have shown us that there can be at least 5 forms of cytosine within a given genome: cytosine 5-MC, 5-HMC, 5-FC, and 5-CAC. One argument for their functions would be that they are intermediates for DNA demethylation. As eloquently mentioned in Dr. Jorla's presentation, 5-FC and 5-CAC are subject to base excision repair by TDG for DNA demethylation, and this stands as very true with multiple data pointing to that. On the other hand, if you apply the same possibility of different temporal and spatial abundance in a tissue-specific manner, if you apply the principle to these three types of oxidative modified bases, as we do for 5-MC, then this epigenetic signal complexity would be much more deep layered than we had previously thought. And if we go with this notion, then we're calling for the existence of specific readers of these modified bases with distinguishable binding affinities that translates each of these signals to a distinct biological outcome. And several discoveries of the readers of this modified bases do support this idea. And as an example of the reader, I would like to first start with this set and ring associated domain, or simply SRA domain. In a mammalian system, UHRF1 and UHRF2 have the SRA domains. UHRF1 is known to be associated with DNMT1 for guiding the DNMT1's maintenance methyltransferase activity. And it binds a hemimethylated DNA. So DNMT maintains the methylation pattern after replication by methylating the daughter strand, and it is believed that the SRA domain initially binds the hemimethylated CPG for this purpose. But people didn't know how it binds until 3 independent reports in a single Nature issue in 2007, all of which revealed that this domain is a non-enzymatic protein, flips 5-MC for DNA binding, as shown in the bottom left. The protein folds contain a half-moon shape in which the flip base is positioned, so it became the first example by which a particular DNA base is recognized by base flipping for the purpose of DNA binding only, not involving an enzymatic reaction. Interestingly, more data have been coming out showing that the SRA domain can have different binding affinities towards differently modified bases. So in mammalian system, SRA domain of UHRF2, in fact, has been shown to have slightly higher affinity towards 5-HMC compared to 5-MC. On the other hand, SRA domain has been also found in prokaryotic systems recently, and some of them are part of the restriction enzymes. For example, the SRA domain of the MSPJ1 flips 5-MC to guide its own endonuclease domain to cleave DNA. And interestingly, in bacteria, 5-HMC is not generated post-DNA synthesis as in eukaryotic system, but is introduced to bacterial genome by phage, which generates nucleotides containing 5-HMC that then becomes incorporated into the bacterial genome. So, and there are restriction enzymes such as ABASI that contains this SRA-like domain that looks like SRA domain, whose affinity for 5-HMC is orders of magnitude better than that towards 5-MC. And such affinity differences seem to be determined by the amino acid composition in the binding pocket of the protein where the flip base is positioned. So in further discussing a prokaryotic modified base reader, I want to briefly make a point here. There is another base flipping 5-MC reader called NCRB, N-terminal domain, that has an entirely different structural folds compared to the SRA domain. Like SRA, it binds 5-MC but does not exert any reaction on it. The DNA base recognition by base flipping may have diverse evolutionary origins. But there's an important point to be re-emphasized here, and that is this base flipping framework is distinguishable from typical transcription binding events in which surface residues recognize a relatively long patches of DNA sequence in its intact double helix form. In contrast, the extra helical recognition, such as in the case of base flipping, allows for recognizing a particular base in a variety of sequence contexts across the genome while ensuring a greater degree of exclusivity for the type of base it recognizes. In other words, base flipping minimizes DNA sequence specificity and maximizes the base specificity for DNA binding. And this hallmark probably is the utility of flipping the base for a variety of reasons. So I want to just make a quick note here. As I've been characterizing base flipping as an important mode of DNA binding, I also do need to mention that this is not the only way modified bases are quote-unquote read by proteins. For example, proteins such as MECP2 that has a methyl CpG binding domain, or simply called MBD, can bind methylated CpG dinucleotide without involving base flipping. It engages arginine side chain that recognizes guanine in a bifurcated manner as shown in the left panel. Because arginine's terminal end is planar in resonance, and it can engage in a pi bond-mediated interaction with the methyl group of 5-MC that is positioned 5' to the guanine in the CpG context. So this 5-MC arginine-guanine triad is seen across MBDs as well as more recently in certain zinc finger transcription factors such as KLF4, CFP57, and Kaizo that are known to specifically bind methylated DNA within a very specific and relatively long sequence context. In addition, certain basic leucine zipper family proteins can recognize 5-MC within, again, specific sequence contexts, including human AP1, such as DJUN, and Epstein-Barr virus, BZLF1, also known as a ZTA or Zebra. In these cases, either alanine carbon beta, alanine side chain carbon beta, or the serine side chain carbon beta can engage in a van der Waals interaction with the methyl group 5-MC as shown here in the middle and the right panels. This was a part of my graduate thesis that was recently published in NAR. So clearly there are non-flipping ways to recognize 5-MC, but this usually happens in a sequence-specific context. And the binding affinity for methylated sequence versus unmethylated sequence for these proteins is significantly higher. So here the contribution of methyl signal for the binding, for DNA binding, is within the confinement of specific locations within genomes defined by the DNA sequence to modulate gene expression, perhaps in a different way, which is characteristically different from the mode of DNA binding by the base flipping proteins. In addition to 5-MC triggers, there have been some recent discoveries that suggest 5-CAC could also be read by transcription factors within specific sequences. This scenario also involves target-based arginine-guanine triad, but sometimes there is an additional residue that allows the direct recognition of a carboxyl group of 5-CAC, such as in WT1 transcription factor to the left, and sometimes it doesn't, as in the case of MAX, which is related to CMYC. In either case, 5-CAC has a similar or slightly better binding affinity as unmodified cytosine for the position concerned, but its binding affinity is far better compared to other types of modifications such as methylation or hydroxymethylation. These discoveries point to the possibility that TAT-generated bases may not be mere intermediates for DNA demethylation, but may serve as a distinct epigenetic signal. Next, I'd like to discuss this as erasure of epigenetic modification, as touched upon in the previous presentation by Dr. Grohead. In many ways, the erasures of modifications are under the same burden of activity requirement as for the writers of DNA modifications. If you consider the situation in which a single base out of all possible bases throughout the genome within a variety of DNA sequence contexts needs to be modified, then base flipping becomes an excellent framework to minimize the DNA sequence specificity and maximize the base specificity for the task. And the same framework is again applicable for the erasers. The erasers need to find a modified base that can be located anywhere in the genome, and they need to erase a particular type of base with less room for errors in picking the wrong base to ensure the accuracy of the task. Otherwise, it will generate unnecessary DNA damage. And we already know that DNA glycosylases, which are basic excision repair enzymes, come into the picture here. In this slide, TDG has evolved to do the task of removing 5-FC and 5-CAC for DNA demethylation, in addition to its traditionally understood DNA repairing task of removing the base mismatched with guanine, such as uracil or diamine, hence the name diamine DNA glycosylase. One may then ask, why erasing modification is so complicated, as the initial establishment of those modifications only requires a single-step reaction, whereas basic excision repair needs a lot of processing involved after following the glycosylase activity. And there are certainly other pathways of DNA demethylation that have been proposed, that have been hypothesized, that does not involve basic excision repair. But you may also want to consider that DNA glycosylases are primarily known as DNA damage repair enzymes that have evolved variously and extensively to deal with all kinds of damaged DNA bases that occur in both normal and abnormal settings. And this evolutionary history may be reflected in the structural diversity of DNA glycosylases as shown in the bottom. There are at least 6 different kinds of DNA glycosylases according to the structural folds, and pretty much all of them flip the target base for the excision reaction in order to repair it. Again, base flipping makes sense in this case because DNA damage can happen in a variety of DNA sequence contexts. And when you repair something, you have to make sure that you're repairing a damaged base, not a normal base. And since nature has already developed a whole set of arsenal for recognizing all kinds of unconventional bases, it may have been easier to use this already well-established infrastructure to repurpose one of its enzymes for a DNA demethylation, in addition to the DNA damage repair function. TDG happens to be the chosen candidate, or perhaps there may be another one that can recognize 5-FC and 5-CAC perhaps. But on the other hand, you may ask about what about 5-MC and 5-HMC? There are DNA glycosylases that may recognize them. And as mentioned in the previous presentation, the discovery of TET and TDG was partly driven by initial efforts to identify 5-MC erasers. And 5-MC DNA glycosylase was definitely a candidate for search at the top of the line. However, in mammalian systems, such DNA glycosylase has not been really identified. But as Dr. Gorat mentioned, the glycosylase known as repressor of silence 1, or ROS1, and its paralogs in plants were discovered in 2002 in Arabidopsis that are now firmly established as the bona fide 5-MC DNA glycosylase, which also has some weaker glycosylase activity on 5-HMC in vitro. Although the presence of 5-HMC in plants could be a bit controversial. But so I actually studied this protein briefly for my thesis as well to understand how it removes 5-MC and 5-HMC. And ROS1 is a relatively large protein with at least 3 domains: the N-terminal domain, the C-terminal domain, and the glycosylase domain that is split into N-terminal and C-terminal compartments, the term GDN or GDC, with a long linker in between. Initially, I was focusing on understanding the glycosylase domain, which is homologous to bacterial endonuclease 3 DNA glycosylase that is known to remove damaged pyrimidine bases. Clearly, the base excision function of ROS1 comes from this domain because if you mutate this catalytic residue within this domain, the mutation results in killing the activity. But what about the other domains? So according to other studies, the N-terminal domain is involved in non-specific unit binding, and the C-terminal domain is of unknown function, actually. And what I have noticed in my study is that the portion N-terminal to the GDN, as well as the linker between GDN and GDC, are not essential for the 5-MC and 5-HMC excision activities. So I generated 3 constructs here, one containing both GD and CTD in a single protein termed ROS1 delta N. another containing GD alone and the other containing CTD alone. And I subject the purified proteins from these constructs for DNA glycosylase activity assays, as shown in the gels in the bottom. And this is the DNA cleavage assay that tests the presence of DNA glycosylase activity on fluorescently tagged oligonucleotides containing either 5-MC in the top of gel or 5-HMC in the bottom gel. So the sign as to the right side of the gel denotes substrate and P denotes the product. So the bands running below represent the presence of the excision activity by the incubated protein. The stronger the intensity of the band in the bottom compared to the top, the more active it is. And looking at both gels, the first lane is the negative control, the second lane is a substrate incubated with the GD only, and the third lane is a CTD only. And the fourth lane involves the incubation of both GD and CTD proteins purified separately. The last lane is using ROS1 delta N as a positive control. And if you look at the result, GD or CTD alone does not have any detectable 5-MC or 5-HMC glycosylase activity. But when the two doses are put together, there is a partial restoration of activity that is comparable to the activity generated by ROS1 delta N positive control. Therefore, the data really suggests that the C-terminal domain is modulating the 5-MC and 5-HMC erasing activity. And this paradigm is somewhat distinctive to the idea that a single base flipping agent, such as DNA glycosylase domain alone, is sufficient for the epigenetic erasure activity, as in the case of TDG. In contrast to this, in case of the ROS1, the C-terminal domain is required and facilitates the recognition of the substrate by the glycosylase domain for DNA demethylation. So there's something different about ROS1 that allows this substrate to be recognized and be removed. Unfortunately, the structure has not been solved, so we do not know exactly how this works, but a future study into this would be nice. So in conclusion, despite inherent structural and mechanistic diversity, the writing, reading, and erasing DNA modifications all engage in the base flipping framework to effectively achieve low sequence specific and high base specific interactions with DNA, which is an essential criteria for establishing, maintaining, and regulating the highly dynamic epigenome. And with that, I would like to thank Dr. Zhu from Purdue University, who has provided the ROS1 construct. I would also like to thank Dr. Speck at Emory University, who has provided the construct for Epstein-Barr virus BZLF1 protein. I would like to thank the Chang Lab member, Dr. Hashimoto, who has done a lot of work. A lot of the proteins that I've presented today and been my mentor in the lab, taught me the basics of biochemistry and a lot more than that. And then I would like to also thank Dr. Horton for really providing me with an excellent training with the crystallography and being a good friend. I'd also like to thank Dr. Zhang, who has really helped me with the cloning and has given me a lot of tips throughout my PhD training. And last but not least, I'd like to thank my advisor, my PhD advisor, Xiaodong Chang, who is now at the UTM, the Anderson Cancer Center, in the Department of Molecular and Cellular Oncology. He's a pioneer in this field. A lot of the work that all the work that I've just presented and summarized has been is done under his laboratory. And really, would like to thank him for inspiring me scientifically and guiding me through the doctorate degree program. And with that, I'd like to thank the audience for listening, and thank you very much.
Thank you, Dr. Hung. We've received several questions from our audience, so let's jump in. Dr. Drohat, the first question is for you. Has enzyme-catalyzed decarboxylation of CAC been explored?
It has been explored, and let's see, there are reports of decarboxylation. Just checking some notes here. I mean, so the idea that you can decarboxylate thymine to uracil is well established, and that suggested the idea that that cells might also be able to decarboxylate CAC to give cytosine. So such activity was reported by the Carrel group in mouse embryonic stem cells. But to date, no enzyme has been identified that possesses this activity. So it's really kind of an open question. There remains the possibility that there could be an enzyme that mediates that process, but the enzyme has yet to be discovered.
Thank you, Dr. Drohat. This next question is for you as well. During mammalian aging, there appears to be a net hypomethylation. Can you speculate what is driving the active demethylation pathway during aging?
Good question. I mean, it may be that there is kind of an uptick in TET activity generating substrates, FC and CAC, for TDG and base excision repair. But other than that, I don't have any specific thoughts on it. I think it's an important question, but I don't have any other thoughts than that.
Great. Thank you, Dr. Hong. Do you have anything to add?
I guess that's a question that's that's open to different possibilities. And certainly, maybe an oxidative damage that's produced through the aging process may interfere with the maintaining DNA methylation patterns, but that's just my speculation.
Thank you, Dr. Hung. This question is for you. In your opinion, is base flipping a reasonable target for biopharmaceuticals to target epigenetic diseases?
That's a great question because it's a really nice framework of being able to have some sort of a base specificity. And so the enzyme, a lot of this protein that flips the base has a very specific pocket that's very specific for a particular base. So there's a lot of, I guess, room for the selectivity there in terms of either blocking the protein or modulating its activity. But and as far as its utility for a variety of different applications, I think that's an open question.
Thank you, Dr. Hung. Dr. Drohat, can you give a brief introduction to the structural effects of the variety of epigenetic marks you mentioned?
So the structural effects on DNA itself?
Yes.
Okay. There have been some studies along these lines. There was one report that if you have multiple formal cytosine modifications, so I think it was 3 CPG sites adjacent to one another with all of the cytosines modified to FC, there was a significant effect on overall DNA structure, I think a bend in the DNA. But I think that the idea that if you've just got single modifications, at one CPG site, and just one of the 2 cytosines in that CPG modified, the effects on DNA overall are really pretty minor. I think some single molecule studies suggest that the DNA may be more flexible at sites of formal cytosine modification. But in general, the base pairing is very much similar to Watson-Crick base pairing, compared to FC and guanine base pairs and CAC guanine base pairs being very similar to GC base pairs with regard to stability, and so there don't seem to be dramatic effects of these modifications on DNA structure overall, other than the exception for the clustering of FC modifications, as I mentioned at the beginning.
Thank you, Dr. Drohat. This question is for both of you. I will pose it to Dr. Hong first. Is viral DNA methylated at CPG islands? And if so, by what mechanism?
So the Epstein-Barr virus, in fact, or the other kinds of herpes virus that are known to be methylated as it enters into human cell and into the nucleus, and that's a way of its dormancy. So that's the way it establishes latency. So yes, it gets methylated, and the viral genome gets methylated.
Do you happen to know about the mechanism of that methylation? Is it similar to?
So it's likely that it's methylated through the host DNA methyltransferases, and that targets the viral genome and methylates it. I don't think that the virus itself has its own methyltransferases.
Thank you, Dr. Drohat. Do you have anything to add?
No, I don't think I would add anything to that.
Okay, great. Well, that was a wonderful webinar. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers directly. Their email addresses are displayed on the screen. As a reminder, the webinar will be archived on the Scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today for sharing your questions and comments. On behalf of the Scientist, I'd also like to thank our speakers, Dr. Drohat, Dr. Hong, as well as our webinar sponsors, BioLegend, Biotics, Active Motif, and Zymo. Thank you, everyone, and goodbye.